Our first presenter is Haley. And I have to say, Haley is not always, we always be remembered at Max Planck for her smile. She's always in a good mood. But also what strikes me about her is that we receive a lot of applications, and I will say at least 50–60% is kids that like, oh, I want to be a neurosurgeon, I want to be another kind of surgeon. Haley was the one that write, I love molecular biology and I love genomics. And like for being someone who has a degree in genomics, I'm like, that is fantastic, but still enters the problem. We need to find the right project for her. And actually we had a molecular biology project, so it was really a match made in heaven with the Yasuda lab. And she did a great work in 6 weeks. So Haley?
Hi everyone, my name's Haley Garcia and I'm a rising senior at Palm Beach Central High School. This summer I've worked in the Yasuda lab under the supervision of Dr. Ogawa on expanding an efficient DNA barcoding system in the Synapse. So, the Yasuda lab mainly focuses on neurotransmission in the synapse. Neurotransmission is essentially the way that two neurons communicate using chemical signals. So, the signal will go from the presynaptic cell right here through the synaptic vesicles to the postsynaptic cell in order to communicate. One example is that when you want to move your leg, the neurons in your cerebellum, which is the part of your brain that controls movement, will send signals through the neurons down to your leg in order to move your leg. However, it isn't as simple as just flipping a switch or typing in a small code. There are thousands of proteins within the synapse that control this neurotransmission and control other functions. So right now, the only way we can see the synapse is through microscopy, specifically fluorescence microscopy. We can see the synapse through electron microscopy, however, we wouldn't be able to view live samples, so we want to use fluorescence microscopy instead. However, fluorescence microscopy has a limited resolution, so it's difficult to see individual synapses and synaptic proteins. If you're not familiar with neuroscience, you may not even be able to really tell where the synapses are. Fluorescent microscopy also is limited to a few colors, so we wouldn't be able to efficiently tag and fluoresce all proteins in different colors since there are thousands of proteins in the synapse. We want to be able to see these synaptic proteins and quantify the synaptic proteins because every neuron in our brain and body communicates through neurotransmission, and essentially all functions in our nervous system are controlled by these synaptic proteins. Synaptic proteins also control synaptic plasticity, which is how neural networks change over time to modify their connection strength. This overall is thought to control memory and learning, which is also heavily studied in the Yasuda lab. In addition, since synaptic plasticity and neurotransmission occurs in all parts of the brains, it is also thought that we'll be able to better study neurodevelopmental diseases and neurodegenerative diseases by viewing these synaptic proteins. So, in order to quantify these synaptic proteins, my mentor came up with a method of DNA barcoding identification by next-generation sequencing. Essentially, how this would work is a DNA barcode would be ligated to an antibody, which would then be incubated with a bunch of cells. It would then attach to the antigen, or the target protein, that we want to quantify. Then we will count the amount of barcodes using next-generation sequencing. I'll break this down a little bit more for you, though. A DNA barcode is just a short sequence of base pairs. You're probably familiar with DNA. Everyone has DNA. DNA is made up of base pairs, adenine, guanine, cytosine, and thymine, to create specific proteins. But for our purposes, we're going to use just a few base pairs to create these DNA barcodes. Then, once it is attached to the protein, we'll run it through next-generation sequencing. Next-generation sequencing is a database with millions of base pair combinations. So essentially, we'll run it through, identify the specific barcode we want for that specific protein. The amount of barcodes we're able to identify is effectively the amount of proteins which is within that synapse. However, my goal for this project was a little bit more specific than my mentor's goal. My goal was to efficiently produce a nanobody to target specifically the HOMR1 protein. In order for this project to work, we use immunolabeling. Immunolabeling is when you use an antibody to target a specific antigen. In this case, the antigen would be the HOMR1 protein that we are targeting. However, a typical immunolabeling will not work for this project. Typical immunolabeling uses the immunoglobulin antibody seen right here. However, the immunoglobulin antibody is 150 kDa, which is too large for us to properly penetrate the synapse. In order for this to work, we want to use a nanobody, which is about 1/10 of the size. 15 kDa. These nanobodies are also much more temperature resistant and have a much better penetration depth, so we can more efficiently reach the proteins that we are targeting within the synapse. In order to produce these proteins, we're going to use the PURE system, which is a recently developed technology to create proteins using in vitro transcription and translation. This method uses E. coli and solution mixes that are provided in the kit, and all that you need to do is provide the target DNA that you want for your protein to be produced. In this case, we're going to take the DNA that we want from a pre-existing Homer 1 antibody. What we're going to do is we're going to make this antibody essentially much smaller. So the antibody's base pairs currently is around 5,000 base pairs. That's going to be way too large for us to use. So we're going to use 2 primers using PCR to effectively take a short region of DNA that we want to amplify. PCR stands for polymerase chain reaction and efficiently multiplies and amplifies DNA using a system of heating and cooling down. For our purpose, we're going to be amplifying a region that is around 439 base pairs. After this PCR is performed, we can compare it to a DNA ladder on an agarose gel. Efficiently, what this does is just show how many base pairs are in our sample to check that the DNA properly produced. In this case, the DNA ladder here shows different notches that mean different amounts of base pairs. Here is the notch for 500, and since we wanted a region that is 439 base pairs long, we can see that the band is around that area and that we efficiently amplified the DNA we wanted. Then we will do a second PCR, which effectively does a very similar thing and is the same process. However, during the second PCR, we're going to attach a promoter, a histidine tag, and a ribosome binding region, which will become useful in the future when we use the PureForX method. This will make the DNA a little bit longer at around 510 base pairs. Then we'll check it the same way using the agarose gel. And you can see here that the band is around 510 base pairs, so it did properly amplify. In this case, the band is a little bit lighter, likely because the concentration was a little bit lower, but it can still be used in the PureForX method. Then we will put the DNA that we just created in the PureForX method that I previously mentioned at the beginning of this presentation. Then we'll run it through a protein gel, which is essentially just for us to check that the nanobody was actually produced. We did the Homer 1 nanobody in 2 different conditions. They were essentially a little bit different. One was just to see if the nanobody would express better. We decided to use condition 1 because, like I said previously, we wanted the nanobody to be about 15 kDa. Right here's the band for 15 kDa, and when you look over, you can sort of see a little band right here. It's difficult to see, but we're able to check it in a different software processing program to ensure it's there. Anyway, we proceeded with condition 1. Then we moved over to a His Gravitrap Talon column in order to further purify the nanobody and ensure that we get all the excess products of the PureForX out of the nanobody. This is done through a process of binding and elution buffers. Essentially, what the binding and washing buffers do is just unattach these excess products, and the elution buffer will keep just the nanobody in the filter, so that's what we'll use in our final product. After we produce the nanobody using PureForX, we can check the nanobody using fluorescence microscopy. You may be confused because at the beginning I said fluorescence microscopy was not going to be suitable for this project. However, it is suitable for us to check the nanobody is actually working and expressing. So what we'll do is we're going to use 3 different samples to ensure that the protein is fluorescing. In this case, we use an anti-PSD95 nanobody, which my mentor has already done and expressed. As you can see, this was tagged with Alexa 488, so it fluoresced green. You can see the green here and see the nanobody. Same thing with the anti-Homer 1 antibody, which fluoresced green in this image. However, the anti-Homer 1 nanobody, which I produced, did not fluoresce green. We believe that this is likely because of a concentration issue, and in the future, we hope to continue this experiment and replicate the results so it will fluoresce. In conclusion, at my time here at Max Planck, I efficiently produced a nanobody for Homer 1. Although it did not fluoresce, the nanobody was efficiently produced, and our results can be replicated in the future, so it hopefully will fluoresce. We also, in the future, we hope that the barcode can be ligated to this nanobody, so it can be efficiently quantified by next-generation sequencing. Unfortunately, this project took a very long time because PCR can sometimes amplify regions you don't want, and there was also a lot of problems with the actual experiments themselves where the concentrations became too low. Like Erica told me earlier, molecular biology is basically just mixing a bunch of clear liquids and hoping for the best. So we had to hope for the best a lot. But if we can efficiently produce nanobodies with DNA barcodes for all proteins in the synapse, we can effectively change the study of the synapse forever and possibly study the brain better as a whole. I would like to thank the Yasuda Lab and Dr. Yasuda so much for having me during my time here at MPFI. And I would like to extend a special thank you to Ira for always being there for me in the lab. Also, thank you so much to Ty for being one of the best mentors I could ever have and teaching me everything about molecular biology that I can carry with me through the rest of my career. I would also like to thank MPFI Education Outreach, Alara and Erica for providing me with this opportunity, as well as my fellow mentors who were always there for me when my experiments failed. And most of all, I would like to thank MPFI donors and supporters for allowing me to be here. I will now open the floor to questions.
When I say it is a real internship, I mean it. Like, we don't have anything planned, so in real science, things sometimes don't work at the first try. Question for Haley.
Yeah, you should stay with me. Actually, I wanted to thank you. I can't tell you the number of times people have tried to explain to me about the DNA coding in the various viruses. In the various samples. I've never understood it. I finally think I've got it. I've got a law background, so thank you.
Of course.
Any other question? Great job.
Thank you.